The overall goal of this experiment is to utilize CRISPR-Cas9 gene editing to model Calreticulin mutations in cytokine-dependent hematopoietic cells in order to study their oncogenic activity.
This method can help answer key questions in the hematological malignancies field, such as the biological function of driver mutations when they are expressed at physiological levels.
The main advantage of this technique is that it utilizes the CRISPR-Cas9 gene editing system to model and study the biological function of genes involved in cancer.
To anneal and phosphorylate the oligos, add the reagents to a total of 10 microliters in a PCR tube kept on ice. Set the program in the thermocycler. Use water to dilute the reaction after cycle is complete. To digest the Lentigard purovector, prepare 50 microliters of digester reaction. Then, start the digestion reaction at 55 degrees Celsius in a heat block. After 1 hour, add an additional microliter of BSM-B1 restriction enzyme and continue the reaction for another hour. To dephosphorylate the cut backbone, add 7 microliters of phosphatase reaction buffer and 2 microliters of phosphatase enzyme to the digested vector. Then, incubate the mixture at 37 degrees Celsius for 30 minutes. Purify the already cut and dephosphorylated backbone using a commercial kit.